Episode 9, our final episode on the digestive system, with a focus on the processes of digestion and absorption. Up until this point, we've highlighted the primary and accessory organs of the digestive tract. And in this episode, we'll now turn our attention once again to looking at how exactly we can break down and absorb nutrients. So on this particular slide, we highlight the role, particularly of the small intestine in absorption. And owing to the large surface area with the circular folds, villi, and microvilli, as we've highlighted in preceding lecture episodes. Now, absorption occurs through a series of mechanisms, including with water, and that happens via simple diffusion or osmosis. There are other substances, though, that are absorbed through more complex mechanisms. We have, as an example, sodium can be absorbed via secondary active transport, and glucose can be absorbed via sodium cotransport or coupled transport. Now, notably, there are different ways and time points at which substances get absorbed. And so we'll take a look at that in more detail on the upcoming slides. So on figure 23.28, what you can see is the breakdown of mechanical and chemical digestion, as well as absorption of the various substances that we typically intake. Notably, most absorption, once again, occurs in the small intestine. This is against popular belief when we think about the stomach. Now, the stomach plays a role more so in mechanical and chemical digestion, as you can see highlighted here on this particular figure. The only substances that typically get absorbed in the stomach include alcohol and aspirin. This is a stark comparison to the absorption of peptides, amino acids, glucose, fructose, fats, water, minerals, and vitamins, which get absorbed in that small intestine. You have a further absorption of ions, water, minerals, vitamins, and organic molecules, which can occur in the large intestine. So this is just a very nice summary and breakdown of, once again, mechanical, chemical, digestion, and absorption. Let's take a look first at the digestion of protein, and this was highlighted in figure 23.30. The digestion of protein begins in the stomach and is completed in the small intestine. Of particular note, hydrochloric acid and the activated version of pepsinogen, known as pepsin, both play a role in the digestion of proteins in the stomach. As the proteins exit the stomach and make their way into the small intestine, there are protein digesting enzymes that are secreted from the pancreas into the small intestine. Once again, this being a major site of protein digestion where final digestion will occur. A small amount of dietary protein is then lost through the feces. You may recall it is the proteases that are the enzymes which help to break down proteins. Proteases are known to catalyze the hydrolysis of proteins into intermediate compounds, and then finally into amino acids, which are those basic building blocks that we know typically lead to protein formation. The main proteases, one of which we've highlighted already, is pepsin, and that comes from the gastric juices present in the stomach. However, you also have trypsin and chymotrypsin, which are contained within the pancreatic juices and meet up with the proteins in the small intestine. Notably, it is these different types of proteases which will catalyze the breaking apart of very specific kinds of peptide bonds that exist between different amino acid combinations. Here is a sample flow chart highlighting the digestion of protein. Once again, the breakdown of protein starts with a protease such as pepsin, which helps to break down the protein into larger polypeptides, which then subsequently get broken down into intermediate and then um, single amino acids, which are those basic building blocks of proteins. So that summarizes once again the breakdown of your proteins. Now on to the breakdown of carbohydrates. Notably, your polysaccharides are hydrolyzed by amylases. This begins in the mouth with the release of saliva, but can also occur with the release of pancreatic juices into the small intestine. What this essentially does is helps to break down carbohydrates as polysaccharides into disaccharides. Disaccharides are able to bind onto various enzymes such as sucralase, lactase, and maltase at the surface of the brush border and undergo additional digestion to form monosaccharides, mainly glucose. Glucose is one of those primary end products of digestion and is conveniently located at the site of absorption. As mentioned earlier, sodium cotransporters, which are a form of secondary active transport, can aid with the absorption of glucose. This process with sodium cotransport of glucose is driven by the movement of sodium ions out of the epithelial cells by primary active transport. This flow chart once again highlights the breakdown of polysaccharides into disaccharides and monosaccharides, owing to the enzymes such as amylase, maltase, sucrase, and lactase, as mentioned earlier. One of the primary, once again, primary end products of carbohydrate digestion is glucose, which then can be absorbed. And now this brings us to fat absorption or lipid absorption. Unlike amino acids and simple sugars, lipids are transformed as they are absorbed through epithelial cells. You may recall from our earlier lecture episodes that the breakdown of fats requires bile. Most nutrients are absorbed by transport mechanisms at the apical surface. However, excluded from this includes your lipids, fat-soluble vitamins, and most water-soluble vitamins. With the help, therefore, of bile, and in particular bile salts, what happens is that the dietary fats can be emulsified or broken down to form small sphere-like structures that are known as micelles, which can then carry fat particles to the surface of the enterocytes. It is at the enterocytes where the micelles will actually release their fats to diffuse across the cell membrane. The fats can then be reassembled into triglycerides and mixed with other lipids and proteins that can pass into lacteals, as highlighted on this diagram. I should also note that at the brush border, as we can see here on this screen, that simple lipids released from the micelles will actually cross the brush border via simple diffusion. It is at this point that the simple lipids then go on to form the triglycerides. The triglycerides are then packaged by Golgi apparatus, as highlighted in the middle of the diagram, into water-soluble chylomicrons, which are then transported either in the blood or in the lymph via lacteals, which make their way also into the systemic circulation. This process is further summarized on this screen here with the flow chart, starting at the small intestine with the emulsification of dietary fats. The breakdown into more water-soluble droplets enables the spherical shells, known as your micelles, to actually help bring the fats uh, and make them accessible for absorption. Notably, pancreatic lipases are enzymes which help to hydrolyze or break down these lipids that are released from the micelles into very simple lipids, which then can be absorbed. Undigested lipids will be eliminated in the feces.